Hello, you are listening to 1 in 2, a Manchester Cancer Research podcast, brought to you by the University of Manchester and the Manchester Cancer Research Centre. With 1 in 2 of us receiving a cancer diagnosis at some point in our lifetime, it has never been more important for our research to improve the outcomes for people affected by cancer. I am your host, Sally Best, and throughout this series, I will be speaking with Manchester cancer researchers about their innovations, discoveries, and projects that are changing the landscape of early detection. In this episode, we speak to Dr Marilena Hadjidemetriou about her research on nano-omics, which aims to integrate nanotechnology to the world of omics, as well as proteomic biomarker discovery. We find out how nano-omics is being used to isolate molecular biomarkers that highlight the early stages of cancer in patients, making earlier cancer detection possible. Marilena, hello! Thank you so much for joining me. How are you doing?
I'm very well, thank you.
Well, I'm wondering if for the sake of our listeners, you'd be able to just introduce yourself, what it is that you do, who you are.
All right, so my name is Marilena Hadjidemetriou, and I am a lecturer at the University of Manchester, leading the Nano-omics team.
Oh, well done. And I'm so glad you pronounced your second name, not me. That would have been absolute carnage. So you currently work with Nano-omics, and it's your team that introduced this term. Would you be able to just explain what it is for me and the listeners? Because, yeah.
Yes, of course. So Nano-omics aims to integrate nanotechnology to the world of omics. It can be defined as the nanotechnology enable isolation of molecular biomarkers from blood or other biological fluids for their subsequent omic analysis, and this could be the analysis of proteins, lipids, or genes. In other words, nanoparticles, and these are materials that have at least one dimension in the nanoscale, so between 1 to 100 nanometers, are utilized as scavenging platforms to capture, enrich, and isolate novel disease-specific biomarkers from biological fluids. For example, we have developed a nano-omics pipeline for protein biomarker discovery, which allows us to see deeper into the blood proteome, and this is the entire set of proteins that are present in blood. This is extremely important for the discovery of novel blood-based biomarkers.
Okay, so just to clarify, absolutely tiny things in your blood that you're then pulling out of the blood, maybe amplifying, and you trace them. So it might be lipids, proteins, genes, and you're looking for cancer signatures.
Yes, exactly. So blood contains a treasure trove of information. We just need to find ways to access this information.
Okay, cool. Just trying to work it out in my head, but I mean, it sounds very complex, but we're gonna, go into it more and work it out. So, I mean, at this point, please could you explain what a biomarker is? Because I know you've mentioned it before, and a lot of our audience might not be aware of that.
So the term cancer biomarker covers a range of biological molecules that are present in blood or other biological fluids, including proteins, genes, and lipids. These are molecules that can be objectively measured in order to diagnose or guide treatment of various diseases, including cancer.
Okay, so basically cancer signatures is a way to snip it.
Correct.
And you've mentioned this nano-omics proteomic pipeline. Do not get me to ever say that again, ever. But I mean, it sounds very impressive. And again, could you just explain a bit more in detail what this is and how it works?
So proteins are the most commonly used blood biomarkers, although a number of protein biomarkers are used clinically to monitor cancer progression. Currently, there are no biomarkers used for cancer early detection. And one of the biggest problems in protein biomarker discovery is that highly abundant proteins, such as albumin, that are blood circulating, are masking the identification of these smaller and lower concentration proteins, some of which are secreted by cancer cells or in response to cancer progression. And so due to this masking effect, the identification of these smaller and rarer proteins is extremely difficult. So we cannot obtain high-resolution protein signatures.
Okay.
In nano-omics, we employ nanoparticles as scavenging agents to pick up these blood-circulating protein molecules of low concentration. This allows us to detect subtle differences in the blood proteome of cancer patients, which could be further developed into a blood test for either early detection or disease monitoring.
Okay, so basically we've got a big ball pit, we've got massive balls, and then we've got tiny balls. Can't find the tiny balls, and we're going to use the analogy that the proteins that cancer cells produce, they're the tiny balls, and then you've got these big ones that are just circulating in your blood. Am I right?
Exactly, yeah.
Okay, next thing, a small child, we're going to call that a nanonet.
All right.
The nanonet goes in and collects up and amplifies all the tiny little balls, and that is our thing that gives us the signature of whether there's that cancer present or not.
Yes. So when you fish, you can either collect buckets of water, hoping you will catch this small and rare fish, or you can throw a net. In the same way, we use nanoparticles as our nanonets. Okay. We immerse them into the blood sample, and then we allow them to interact with the blood circulating proteins. We then lift these nanonets and analyze only the proteins that are here onto them.
That analogy was a lot better than mine. Fab. I mean, okay, so that is a really good analogy, and I can really picture it clearly, and I think it's one of those things that is important for our listeners, because a lot of us might not function on this kind of submolecular level, so understanding things at that kind of bigger picture of a trawler and layers of large fish and the tiny little ones that we want to know about because they are that cancer signatures. So I'm just wondering, just to clarify, the only proteins that adhere onto these nanonets are the ones that are secreted by cancerous cells.
No, many different types of blood proteins interact with the surface of nanoparticles. There is no specificity towards cancer-secreted molecules. However, by analyzing this subset of blood proteins adsorbed onto the nanoparticle surface, and by purifying any unbound proteins, we are able to search deeper into the blood proteome and identify small and low abundant proteins, some of which are secreted by cancerous cells. By then comparing the blood samples at healthy and disease states, we can identify those proteins that are either upregulated or downregulated in response to the disease. And these are the potential biomarker proteins.
Okay, so there's a lot of post work that you have to do post the kind of nanonet retrieval. And I mean, we focused in the season on the early detection of cancers, and I'm wondering, specific to this work, what is the role of nano-omics in the early detection of cancer?
We know that detecting cancer early increases the chances of survival. And so Nano-omics' ultimate goal is to use nanotechnology in order to uncover biomarkers in blood that can be used for early cancer detection. This could allow patients with cancer to receive effective treatment before tumor burden becomes incurable.
Okay, so yeah, detecting early from blood samples. I mean, as well, it's not kind of as invasive as a lot of the tests that you might use in general kind of cancer detection and
Exactly.
things.
It's much less invasive than tumor biopsy, for example.
Yeah. And a biopsy, just to clarify, is cutting out of internal and then monitoring and looking into it.
Yes, exactly.
Okay, cool. And I mean, this all sounds incredible. Do you have a real world example of this and like a key project that you're working on that we can kind of tell to the listeners so they get an idea for, you know, what sort of things you're working on in the background?
We're currently privileged to be working with Professor Caroline Dive and Dr. Phil Crosby, looking into nano-omics and biomarkers in lung cancer. We know that when lung cancer is found at an early stage, patients can often be cured through early intervention. However, when lung cancer is found at the metastatic stage, and this is where it has already spread throughout the body, the prognosis is often poor. And so in this project, we use nanoparticles. These are lipid-based nanoparticles. We call them liposomes, as our scavengers to look for early detection biomarkers. Our aim is to utilize the nano-omics pipeline to analyze pre- and post-surgery blood samples collected within Manchester community-based screening project. And by doing that, we hope we will be able to extract information from blood that in the future can aid in early lung cancer detection and disease monitoring.
Amazing. I mean, and I've had a pleasure of speaking to Phil earlier in the season about lung health checks. So it's amazing to hear about, you know, these other projects that he's working on, and he must never sleep because he's just, always doing things. And I'd like to take a few steps back here because it's something that I've kind of asked everybody else. What led you into this field of nano-omics and cancer?
So the nano-omics idea was generated during my PhD project, and this was like 10 years ago. And the main goal of my PhD was to understand the interaction of nanoparticles with blood proteins. And this is a spontaneous interaction that happens as soon as nanoparticles are exposed to a biological fluid, also known in the nanoscience community as protein corona formation. So during the last year of my PhD, I worked in collaboration with the Manchester Cancer Research Centre and the CRISIS Biobank team, and we were able to recover and characterize nanoparticles from the blood circulation of ovarian carcinoma patients. We recovered a nanoparticle-based chemotherapy drug that is administered in patients with recurrent ovarian cancer. And this was the first investigation of the interaction of nanoparticles with blood proteins in human patients. So we isolated and purified the nanoparticles from the blood of ovarian cancer patients and analyzed the corona that was formed around them. And what we observed was that nanoparticles were able to pick up ovarian cancer-specific blood circulating proteins, which indicates that the composition of protein corona around the nanoparticles reflects the ongoing pathology. This reinforced the idea of utilizing nanoparticles in order to surface capture and amplify cancer-specific biomarker proteins of low abundance. And then during my postdoc, I further developed this into a pipeline for protein biomarker discovery ex vivo, so when nanoparticles are incubated in a tube with samples, plasma samples obtained from cancer patients. And we're now working to further develop this technology for the multi-omic analysis of blood.
Okay, so just to clarify, in this ovarian cancer specific project, your nanoparticle was it was in the blood circulation and it was trying to pick up proteins that were kind of released by ovarian cancer cells. And the early detection angle is because when you first start developing cancer, there's only very low levels of that protein. So then you have to amplify them to higher levels and look at them in more detail kind of outside of the body. Is that right?
Exactly. Okay. You got it right. So this is an enrichment step for those very low concentration proteins. So nanoparticles are fishing these proteins.
Yeah.
And it's a discovery tool. So you use it to identify those proteins, but then you have to develop it into a blood test that will be clinically used.
Okay. And then would that just be like general screening that we'd use that test? So would it be people that would come into the clinic and have screening for, say, lung and ovarian cancer through blood tests?
It could be that, or it could be a companion diagnostic.
Okay. And that would just be would that just be a healthy population of people, or would that be people that had kind of experienced family hereditary links? I know you're not a clinician, so it's kind of a difficult question to ask you.
No, I think it could be both.
Okay. Another question. I'm just so interested in this, but so you've got really low levels at this early stage. Would you then see kind of a massive increase in those levels of biomarkers as you get to stage 4, so that you don't require that kind of amplification level?
You could either see a massive increase or a massive decrease.
Oh, really?
So a biomarker can either be upregulated or downregulated in response to cancer.
God, it's so interesting. God, look at you. Amazing. Honestly. And I mean, just moving on again, you're Cypriot, as we can all hear. What a wonderful accent.
Yes, that's correct.
I am so jealous of both the accent and where you're from. But I mean, what the heck encouraged you to come across here? I mean, I'm kind of presuming it was your research that gave you that, not the weather.
So I'm a pharmacist by training, and I obtained my first degree in Athens. I already knew from the first year that I wanted to do research. That was a 5-year program. And when during the 4th year of the course, I attended a lecture on nanotechnology. And I was immediately fascinated by cancer nanotechnology and the use of nanoparticles for drug delivery purposes. So after the completion of my studies, I decided to move to London, to UCL School of Pharmacy, to obtain a master's degree on carbon-based nanomaterials. This is where I met my PhD supervisor, Kostas Kostarelos, who was about to move from London to Manchester to explore the use of graphene-based materials for biomedical applications. And this is how I ended up in Manchester.
Yeah, and of course we've got a massive graphene centre here. And you're going to stay, aren't you? You're not leaving.
I hope so.
Keep funding, come on. So you've mentioned you're now going to develop this tech for the multi-omic blood analysis tool. Would you be able to explain what a multi-omic tool is and the work you're carrying out on multi-omics?
So we know that a single biomarker cannot provide the sensitivity and specificity needed for early detection. Instead, we need panels of biomarkers consisting of multiple molecular classes, including proteins, lipids, and genes. Currently, there is no other technology that is able to offer multi-omic analysis of blood with a single enrichment step. And this is the blood analysis gap we seek to fill. So what we've realized over the last couple of years is that nanoparticles not only interact with proteins when incubated in blood, but can also interact with other biological molecules, such as cell-free DNA and lipids. And therefore, what we're trying to do is to develop a nanoparticle platform that will simultaneously enrich the cancer-specific proteomic, lipidomic, and genomic information with a single nanoparticle enrichment step. This not only will significantly minimize the volume of blood we need for our analysis, but it will also result in the discovery of integrative multi-omic biomarker panels.
Okay, so it's a very, very, very selective net that kind of picks out and amplifies very, very selective proteins and biomarkers and lipids and genes and...
It selects a combination of biomarkers.
Okay. God, wow. The things that you guys are doing. Please, could you give me an example of where you're using multi-omics?
So an example of a project we're currently working on is funded by Cancer Research UK and the International Alliance for Cancer Early Detection. This is in collaboration with the Canary Center at Stanford University. And so what we're aiming to develop is an integrative blood biomarker discovery, a proteogenomic platform. This means you integrate proteomics with genomics for the early detection of non-small cell lung cancer.
So you're working with ACE and Stanford. I mean, why? How has this collaboration come about? What are ACE kind of doing with you? What are Stanford doing with you? What's the partnership look like?
So what we're trying to develop here in Manchester is the nanotechnology platform that will integrate the proteogenomic information in the blood of non-small cell lung cancer patients. And our collaborators at Stanford will then use this information to develop integrative bioinformatic analysis platforms.
Okay, and then what's ACE contributing to that?
ACE is funding this project
cool.
for us to build new connections with Stanford University.
Oh, wow. Look at you go. And Stanford as well. Woo. I'd like to look at this and approach it from more of a patient angle. So, I mean, what does this mean for patient treatments and outcomes of these cancer patients?
So such multi-omics early detection approaches hold great potential in providing precise information on the localization and the size of the tumor. We hope that these novel panels of biomarkers discovered will be translated into simple blood tests that will enable cancer early detection, monitoring, and will facilitate the design of personalized therapies without the need of invasive tumor biopsies.
Okay, so it's amazing that it has the specificity to both kind of know the staging and the location of tumours, as well as the fact that it negates the need for tumour biopsies, which is, again, massively kind of invasive, like you've said, and there can be a bit of recovery and distressing and things. So
exactly.
Doing great work. I love it. And I mean, it's a question I like to ask all of our guests. Some of them don't like it, but I love it. So it's, why are you passionate about your research?
I'm passionate mostly because of the multidisciplinary nature of nanomics. We use advanced materials and nanotechnology to address pressing clinical needs. Even though we are based at the main university campus, we work closely with the Manchester Cancer Research Centre and the Christie's Hospital, and this allows us to integrate technology development with clinical research. Manchester brings together a very rich ecosystem of cancer research, and it creates this unique opportunity of clinically translating our technology innovations for personalized medicine.
You came to England and you spoke about, before the podcast, the environment of Manchester, and why you've chosen Manchester to build your research portfolio following your PhD. And I'm wondering if you could just expand on that, give these listeners a little tidbit about what you kind of told us about, the integration and the landscape of Manchester sort of thing.
I think Manchester is a great city for cancer research. And the reason for this is a collaboration between technology development and clinical research. So it becomes very easy for us to get access to clinical samples and collaborate with clinicians. And this is a unique opportunity to see our technologies being translated.
And I mean, you've said you've been given opportunities to move elsewhere. I won't say where because you're not going. What's keeping you here? What's that kind of drive to want to keep your research here?
I think because the Nano-omics was generated during my PhD, I wanted to see how Nano-omics will further be developed into clinical research and cancer research.
Finally, it's been such a nice conversation with you, and there's so much to go into, but fortunately we don't have the time. What are your hopes for the future, like your personal future, as well as the future of nano-omics?
So I hope to see some of these newly discovered biomarkers clinically used. The discovery phase of biomarker development is often initiated in academic labs and frequently results in the identification of multiple biomarker candidates. Translating these discoveries into clinical tests requires tremendous resources for analytical and clinical validation. And this, of course, requires collaboration with industrial partners. I believe we have not yet fully explored nano-omics, as this is a new area of research. And nanomics can be applied across a range of clinical needs and biomarker applications, including early detection, minimal residual disease monitoring, and patient stratification. But what is even more interesting is to explore the application of nanomics in understanding the molecular pathways that are involved in cancer progression. And this could potentially lead to new treatment approaches.
New treatment approaches, and Manchester pioneering, which is stuff that we love to hear. Honestly, thank you so much for taking the time to speak with me today. Absolutely killing it in this research area, and can't wait to find out about all the project updates.
Thank you so much,
keep
Sally.
us updated, please.
Thank
We want to hear about it. Come back again. And I mean, as usual for the listeners, I'll link everything related to Marilena's work in the blurb. But yeah, thank you for listening, and hope you can all join us next time. Bye. If you have been affected by anything you've heard in this episode, please see the show notes for a list of charities and organisations that can help.
1 in 2 was brought to you by the University of Manchester and the Manchester Cancer Research Centre. Listen to our next episode to hear from more of our researchers as they share the innovations, discoveries and projects that are changing the landscape of cancer prevention, early detection and treatment. To find out more about what you've heard today, please see the show notes for this episode, where you'll find a transcript and links to further information and research. Cancer is one of the university's 5 research beacons, showcasing the interdisciplinary collaborations and cross-sector partnerships that are tackling some of the biggest questions facing the planet. To hear more about Manchester's research in advanced materials, biotechnology, cancer, energy and global inequalities, go to manchester.ac.uk/beacons.